food industry carries the primary responsibility for seeing that food produced is safe and will not harm those eating it. One of the best ways to produce safe foods is to understand the major factors that affect food safety and how these can be controlled. Contamination is the presence of harmful substances or conditions in the food that can cause illness or injury to people who eat the tainted food. There are multiple points where foods may become contaminated. Foods can become contaminated during growing and harvesting of raw materials, during storage and transportation of these materials to the processing plant, and during processing. Finished products may be contaminated during storage and transportation to the point of sale, or while being stored or handled by the consumer. Food may be contaminated by microbiological contaminants, chemicals, and physical contaminants. While all of these types of contaminants offer potential problems for a food manufacturer, microbiological hazards, particularly those from microorganisms, are of the greatest concern since they can be the source of foodborne illness and even death for those consuming contaminated food. So, it is especially important that the food industry assume responsibility for preventing risks associated with microorganisms. Understanding the organisms involved and the factors affecting their growth and control is the best way to see that these risks don't occur. Microbiology is the study of microorganisms, living organisms so small that they can generally only be seen with a microscope. There are 4 main types of microorganisms that contaminate food and may cause foodborne illness. These are bacteria, viruses, parasites, and fungi. It is important to understand that microorganisms are all around us. They are carried by humans and animals. They are also very common in the environment and are found in the soil, on plants, in water, and in the air. Since microorganisms can be found just about everywhere, it is easy for food to become contaminated with them. They may get in food from soil, water, animal feeds, and other factors in the production environment. During processing, equipment and utensils, ingredients, and environmental factors like air, dust, and condensation can be sources of microorganisms. While we tend to think of microorganisms as being something we don't want or need, microorganisms perform some important roles for our daily lives. For example, fermentation, which results in the production of many types of cheese, bread, beer, wine, and some sausages, is the result of microbial activity. Microorganisms also aid in digestion of food in the body. They may synthesize a variety of chemical materials, including some vitamins, antibiotics, and certain industrial chemicals. They are important in breaking down waste products and therefore play an important role in waste management and recycling. Unfortunately, microorganisms can also be harmful. Some molds, yeasts, and bacteria cause foods to spoil. Since food contaminated by these organisms is typically considered inedible, it's more uncommon for them to make people sick. Other microorganisms can make people sick. It is important to remember that while there are thousands of types of microorganisms, very few of these are harmful to humans, and there are only a few diseases that are transferred through food. There are about 40 different kinds of microorganisms that can occur in food and cause foodborne illness. These disease-causing organisms are called pathogens. Pathogens include viruses and parasites. However, it is important to remember that while these microorganisms may be carried by food and may cause illness, they cannot grow in food and they do not cause food to spoil. On the other hand, as we've already seen, some bacteria can cause food to spoil. Some bacteria also can cause foodborne illness. For this reason, bacteria are the microbial group of most concern to food processors. Before we consider ways of controlling bacterial growth, let's take a few minutes to consider what we mean when we talk about foodborne illness. A foodborne illness is a disease that comes from food containing a pathogen or the toxin produced by a pathogen. Different foodborne illnesses have different effects on the body. Some cause damage to the cells and may lead to breakdown of body tissue. Some disrupt the processes of the body. And some may cause the body to respond in such a way that it harms itself. There are two main types of foodborne illness: Intoxications and infections. An intoxication occurs when a person eats a food containing a toxin or poison. Although toxins may occur in food from chemical contamination or may even be a natural part of some foods, it is the toxins produced by microbial pathogens which cause the greatest concern for food processors. As shown on the slide, toxins cause foodborne illness by disrupting particular targets in the body, such as the gastrointestinal tract or the central nervous system. Symptoms of a foodborne intoxication vary depending on such factors as the toxin involved, the part of the body affected, and the health status of the infected person. Some toxins are very heat resistant and may be a hazard in food even if it is properly cooked. An infection occurs when a person eats a food containing a pathogen that has been allowed to multiply in the food to a level capable of causing illness in the person eating the food. This level is referred to as the minimum infective dose, or MID. The MID varies for different pathogens and may range from as few as 10 cells for Shigella to as many as 100,000 for Staphylococcus aureus. Infections caused by different types of organisms may have very different effects on the body. Some infections cause acute or immediate illnesses. Some may cause chronic or long-term illnesses like arthritis, impaired immune systems, or birth defects if the affected person is pregnant. The organism Listeria monocytogenes can cause spontaneous abortion of a fetus, while Clostridium botulinum can attack a person's central nervous system. The differing symptoms, durations, and severity of the foodborne illnesses makes diagnosis and tracking of their occurrences difficult. As mentioned previously, bacteria are the microbial group of most concern to food processors. For this reason, let's spend some time looking at bacteria and considering how they might be controlled. Bacteria are single-celled living organisms. They are among the smallest living creatures known, and because of their tiny size, they cannot be seen except under a microscope. They also cannot be smelled or tasted, so detecting their presence in food is almost impossible unless they have caused the food to spoil. Bacteria are single-celled living organisms. They are among the smallest living creatures known. Under the microscope, we see that bacteria occur in several different shapes. The most common in foods are round, called cocci, and rod-shaped, called bacilli. Some bacteria form spores. Spores are a dormant or resting stage of the bacteria that help ensure the survival of the organism in times of environmental stress, such as in periods of extreme dryness or in high heat. Cells of non-spore formers, of spore formers that haven't formed spores, and of spores that have become active again are referred to as vegetative cells. Since spores develop to help bacteria survive adverse conditions, they are capable of surviving in conditions where vegetative cells would normally be killed. For example, many spores can survive heat and cooking and freezing. Generally, spores that are resistant to heat are also resistant to destruction by chemicals. There are some bacterial spores that can survive more than 3 hours in the sanitizing solutions used in food processing plants. These are the same sanitizing solutions that easily kill vegetative cells. Because spores can survive unfavorable conditions like heating, freezing, or drying, food processors must be careful to assure that product handling prevents spores from returning to active vegetative cells. The time it takes to heat and/or cool a product during processing is important. If the product is in a temperature range favorable to bacterial growth for too long, the spores will change to the vegetative state and start reproducing, a state called outgrowth of spores. If vegetative organisms reach the minimum infection dose or are allowed to produce a sufficient amount of toxin, foodborne disease can occur. Some major spore-forming bacteria found in foods are listed on the slide. Because these organisms are present everywhere in the environment and they are difficult to control in food processing, it is important for food processors to know when one or more of these organisms is likely to occur in their products. Bacteria reproduce by splitting in two. So a single cell becomes two become 4, etc. The time it takes for this splitting to occur varies with the type of bacteria and environmental factors, but is usually rapid, usually about 20 minutes or less. As shown on this slide, a single cell could lead to 8 cells in just an hour. Since a food seldom contains just a single cell, it is easy to see that the number of bacteria in a food product can become large very quickly. The time that it takes for a single cell to divide into 2 cells is called generation time, or doubling time. Bacteria have the shortest generation time of all the microorganisms. This is followed by yeasts and molds. For bacteria, this time can be as short as 10 to 12 minutes. Note that the terms bacterial growth and microbial growth refer to an increase in number of bacteria, not in their size. Examples of generation times under ideal conditions at 37 degrees Celsius for some bacteria are shown on this slide. This chart points out the ranges of time a processor may be dealing with when working to prevent bacterial growth, highlighting the importance of making efforts to limit the amount of time foods are held in conditions that might encourage bacterial growth. When we look at the progression of bacterial growth in a food, we see that it can be broken into 4 stages, or phases: lag phase, log phase, stationary phase, and death phase. Understanding what happens in each of these phases helps us understand how to control bacterial growth. First, the lag phase. When bacteria are first introduced into a food environment, they go through an adjustment period called the lag phase. There is very little bacterial growth during this phase. Again, remember that bacterial growth refers to the number of bacteria, not size. This is the most variable phase because the amount of time needed for the organism to adapt to the food's environment depends on factors such as the characteristics of bacteria and the environment. The length of the lag phase affects how quickly the minimum infection dose is reached. The log phase is characterized by rapid reproduction. As we know, bacteria reproduce by splitting into 2, and this is the phase when reproduction is most rapid. With the rapid increase in the number of organisms, nutrients in the environment are used up quickly until one of the nutrients is gone and growth begins to slow, leading to the stationary phase. In the stationary phase, bacteria are growing and dying at about the same rate. Reproduction slows because nutrients were used up in log phase. There is an increase in byproducts from growth that also inhibit further growth, so the number of organisms remains fairly constant. When dying bacteria outnumber growing bacteria, the population declines during the death phase. One of the objectives of food preservation and/or processing is to extend the lag phase as long as possible so that the number of organisms in the product is kept at a minimum. The time required for bacteria to pass through the lag phase and grow in a log phase depends on the conditions in the food. The graph on the slide shows how different temperatures affect the growth of Salmonella species. Salmonella species grow more quickly at warmer temperatures, 95 to 99 degrees Fahrenheit or 35 to 37 degrees Celsius, than at colder ones, 44 to 50 degrees Fahrenheit or 7 to 10 degrees Celsius. At even colder temperatures, 42 degrees Fahrenheit or 6 degrees Celsius and lower, it does not grow at all, although it does not die either, prolonging the lag phase. This is why refrigerating food correctly helps to keep it safe. Like all living organisms, bacteria need specific things to live and grow. Although the levels of these may differ for different types of bacteria, there are 6 basic conditions needed for bacterial growth. Controlling one or more of these conditions will help keep bacteria in food from increasing to dangerous levels and/or from producing harmful toxins. The acronym FATTOM can be used to remember the conditions that must be appropriate for the growth of foodborne microorganisms. These conditions are food, acid levels, temperature, time, oxygen, and moisture. These conditions will be discussed in the next few slides. Food. Every living organism requires food or nutrients to grow. Key nutrients needed by bacteria are protein and carbohydrates. As we saw when we looked at the growth curve, if the food supply is removed or depleted, bacteria will not reproduce. Unfortunately, since bacteria that are in our food are consuming the same things we do, it is difficult to use removal of their food supply as a way to control bacterial growth. Acidity levels, measured as pH, range from a pH of 0, very acidic, to 14, very alkaline. Although the ideal pH for bacterial growth varies with the type of bacteria, all bacteria have an ideal acid level for growth. Pathogenic bacteria typically do not grow in alkaline or highly acidic food. They prefer food that is slightly acidic or neutral, corresponding to a pH of 4.6 to 7.5. The pH of most foods is in the range of 3 to 7.5. Fruits generally have a pH lower than the optimum pH range for bacteria, but most other foods, including meats, milk, vegetables, and cooked grains, all have pH levels that are ideal for bacterial growth. Most bacteria grow best between 41 and 135 degrees Fahrenheit, or 5 and 57 degrees Celsius. This temperature range is called the temperature danger zone, or TDZ. Bacteria grow even more rapidly between 70 and 125 degrees Fahrenheit, or 21 to 52 degrees Celsius. Above and below this ideal temperature range, growth is slowed or even stopped. When the temperature is high enough, bacteria are killed. Extremely low temperatures will slow the growth of the bacteria, but generally do not kill them, so they may begin to reproduce and/or produce toxins when the food is warmed. Food must be handled very carefully when it is thawed, cooked, cooled, and reheated, since it can be exposed to the TDZ during these times. For all bacteria, there is an ideal temperature where they reproduce the fastest, and bacteria are often classified based on this temperature. Bacteria classified as psychrophilic are cold-loving and generally grow best at temperatures ranging from below 0 to 68 degrees Fahrenheit. Most psychrophilic bacteria cannot grow when the temperature is above 77 degrees Fahrenheit. Psychrotrophs are bacteria that grow best between 77 and 104 degrees Fahrenheit. They can grow slowly in and on food at refrigerator temperatures. In Greek, meso means middle and philic means loving, so it is not surprising that mesophilic bacteria grow best in the 86 to 104 degrees Fahrenheit temperature range, the middle of the growth zone for bacteria. Some mesophiles will continue to grow at temperatures up to 116 degrees Fahrenheit. All bacteria that affect food safety are in this group, although some may be considered psychotrophic. Thermophilic bacteria are heat-loving and grow at high temperatures. Thermophilic bacteria may cause foods to spoil, but they are not pathogenic and do not produce toxins, so they do not affect food safety. The more time bacteria spend in favorable growth conditions, the higher the rate of reproduction and the more likely they are to reach population levels high enough to make someone ill. Remember, since bacteria can double their population in a very short time, it is important to limit the time foods are exposed to temperatures favorable for their growth. Bacteria differ in their need for oxygen. Some, referred to as aerobic, need oxygen to survive. Think about why you do aerobic exercise, to get oxygen to your muscles. Some, called anaerobes, cannot grow if free oxygen is present. An is a Greek prefix meaning without, so these are bacteria that grow without oxygen. Still others, facultative anaerobes, use oxygen if it is present but don't require it. These differing oxygen requirements show the importance of knowing the nature of the bacteria you might be dealing with in your food product, since limiting oxygen to control some bacteria might be producing ideal conditions for others. Bacteria get their nutrients by absorbing them through their cell walls. This means moisture must be present to dissolve the nutrients so that they can be absorbed. It is important to note that the moisture must be not just present, but also must be available for the bacteria to use. In some foods, water may be tied up by components of the food, so the food may appear moist, but the water is not available for use by the bacteria. The measure of the amount of water available to dissolve nutrients is called water activity, represented by A_W. The water activity scale ranges from 0, which is no water available, to 1, pure water. Most bacteria need a water activity level of greater than or equal to 0.95 for growth. Most fresh foods have a water activity greater than 0.97. This chart shows the water activity requirements for some of the microorganisms that can grow in food and cause food spoilage or foodborne illness. While most bacteria require a water activity of 0.95 or greater, there are some that can grow when the water activity is as low as 0.91. Yeasts have a lower requirement for available water, and the requirement for molds is even lower. Microorganisms will not grow at very low levels of available water. The water activity of a food can be manipulated so that microbial growth is limited. One way this can be done is by physically removing some of the water naturally present in the food. We do this when food is dried and when it is baked. Another method of limiting water activity is to add ingredients that bind or hold on to the water so it is not available for use by the bacteria. Sugar and salt are two such ingredients. This chart shows the water activity levels of many common foods. As we've already mentioned, most fresh foods have water activities capable of supporting all types of microbial growth. Many processed foods also meet this requirement. Take a minute and look at the foods within the various water activity levels. This has been a brief overview of the 6 basic conditions needed for bacteria to grow and/or produce toxins. Although the levels of these growth requirements may differ for different types of bacteria, it is important for food processors to have a clear understanding of how controlling one or more of these conditions will help keep food safe. So, what are some common ways food processors control these growth requirements? Changing the environment of the food so that it is not ideal for bacterial growth is the most common way food processors limit bacterial growth. For example, since we know that most bacteria will not grow in an acid environment, it follows that bacterial growth can be limited by adjusting the pH of the food to levels that do not support bacterial growth. This adjustment may be achieved by adding an acid, such as vinegar, to the food, a procedure common in many pickling processes. Acidity also may be adjusted by causing acid to be produced in the food. This is what happens in many fermentation processes, such as when cabbage is made into sauerkraut. Controlling the temperature of the environment is another way of limiting bacterial growth. Holding food at temperatures above or below the optimal temperature for bacterial growth may be a way of limiting bacteria. However, as we discussed earlier, different types of bacteria have different optimal growth temperatures, so it is important to know the types of bacteria that may be present in a particular food in order to choose the appropriate limiting temperatures. In addition, the nature of the food product itself, such as whether the quality of the product will be negatively affected by holding it at very high or very low temperatures, may be a factor in selecting appropriate holding temperatures. Other characteristics of the food, such as its pH, water activity, and the presence of ingredients that affect bacterial growth, may also play a role in determining the appropriate holding temperature to limit bacterial growth. Chemicals may also be added to food products to limit bacterial growth. While it is possible to add chemicals that kill any organisms present, not just limit their growth, this is a much less common practice. In order to assure the maximum bacterial control, chemicals are often used in combination with other control methods, such as heat or reduced water activity. This concludes our brief introduction to the microbiology of food processing. We hope this presentation helps you understand the major microbiological factors that affect food safety and how these can be controlled.